So our next speaker is Ann Martin. Ann is a genetic counselor, leads our registry, and has been amazing with PPMD for a very long time. So Ann, I'm going to let you take over.
Great. Thank you, Pat. And thank you, everyone, for joining and spending your Saturday morning with us. I'm really excited that all of you are here and wish we could be together in Atlanta, but maybe next time. All right. Let me move my slide. So my topic isn't quite as exciting as Alpha's. That was really fun to hear about. But my purpose today of my talk, I wanted to kind of give a Duchenne and Becker 101, just a little bit of intro information to lead into the care panel, and then dig into some basic genetics, talk about the importance of genetic testing, and mention our DECODE Duchenne program, which provides free genetic testing. And then I also want to spend some time talking about the Duchenne Registry and how that can help you connect with research and hopefully answer some of your questions. So what is Duchenne? I feel like I'm preaching to the choir here. I'm sure most of you are very familiar with Duchenne, more than you would like to be. But in case there's anybody listening, I just wanted to give a brief background that Duchenne and Becker are muscular dystrophies that are genetic conditions, and they are caused by mutations in the dystrophin gene. So that dystrophin gene, or DMD gene, makes the dystrophin protein, which is essential for our muscle cells to function normally. So without dystrophin, the muscles really aren't able to function or repair themselves as they should. I'm not sure if any of you have seen this diagram before, but I did want to put this in here. This is actually a picture of a muscle complex. It's called the dystrophin glycoprotein complex. It's in the muscle cell membrane. And that's where dystrophin is, and that's where it does its job. The dystrophin is the yellow beaded, almost necklace-like chain that you see there. And basically, the dystrophin helps to really maintain the stability and integrity of the muscle cell membrane. And actually, if any of those proteins are missing from that complex, that can really lead to damage of the muscle cell over time, because the muscle cells, they do a lot of work. They're constantly going through cycles of contraction and relaxation. So without that protein complex there in the membrane providing that stability, the muscle cells can break down and degrade over time, which is what happens in the muscular dystrophies. And that picture there on the right is showing a tire and a shock absorber. And I think it's often easy for people to think of dystrophin as being like the shock absorber. Dystrophin is there to absorb the stress and the movement created by our cells contracting and relaxing all the time. So if you don't have dystrophin, or if your dystrophin levels are much lower than normal, then the muscle cells will become damaged over time and actually become replaced with scar tissue and fat. And that process is called fibrosis. Maybe you've heard of that. The pictures on this slide are showing actually how your muscle cells look under a microscope. So if you've had a muscle biopsy done, this is what they're doing in the lab. They're looking under the microscope and they're staining for the dystrophin protein. So the picture on the left is showing how it would look in a normal, healthy muscle tissue. You can see each cell is outlined brightly with the dystrophin protein, and that is how it should look. The picture to the right is showing muscle tissue from someone with Duchenne, and you can see there's actually no dystrophin present, and the muscle cells do not look normal, and there's actually a lot of spaces between them that are eventually being filled with that scar tissue and fat. So I know we talk a lot about Duchenne being a disease of the muscles, but I wanted to put this slide in to really highlight that it's really not just a disease of the muscles. It affects every system of the human body. And You can see here it affects all of the muscles. It can affect the bones, the heart, especially because the heart, of course, is a muscle. So that is why individuals with Duchenne or Becker or even carrier females can have complications with their heart, because dystrophin is an integral part of the heart muscle. The lungs, our breathing system can be affected, our GI tract or digestion can be affected. Even the brain, dystrophin is actually produced in the brain, and we think that could be leading to some of the behavioral or learning difficulties that we see in some children with Duchenne. And that last picture there on the bottom right is showing the endocrine system and pointing out that even growth can be affected by Duchenne. So our care panel, speaking after me, they're going to touch upon a lot of these different systems that can be affected in Duchenne. So I'm going to focus more on the genetics. I am a genetic counselor, so this is the fun part for me, so bear with me. You're going to feel like you're back in high school biology class, but I do think it's really important to have a solid understanding of the basics of genetics before we start talking in more detail about the gene and different mutations. It can be confusing if you don't understand the basics. But this picture is showing a cell. You know, our body is made up of millions of cells. They're like the building blocks for our body. And the center of each cell is the nucleus, and that's where all those chromosomes are. We inherit the chromosomes from our mother and father, so they contain all of our genetic material and code for how we're going to develop. If we could actually unravel that chromosome, we would see the DNA molecule there. Each little segment of DNA is a gene, and each gene codes for something in particular in our body. So the dystrophin gene or the DMD gene codes for the dystrophin protein. So that's what we're going to be talking about. But of course, there's thousands of genes in our body, in the chromosomes, each coding for something different. If we could even unravel that DNA molecule further, we would see that it's made up of 4 DNA bases. Those are the letters there, the A's, T's, G's, and C's. And maybe you've heard about that or seen those letters on your child's genetic test report. So we'll talk more about that in a minute. So back to the chromosomes, if we could actually look at all the chromosomes in one human cell, we would see that there's 23 pairs or 46 total. The first 22 are numbered, and then the 23rd pair is the sex chromosome pair, and that's important. So women have 2 X chromosomes, where men only have 1 X and then a Y chromosome, because that's what makes them male. The reason I'm pointing this out is because the Duchenne gene is on the X chromosome. So you often hear of Duchenne as being an X-linked condition. That just means that the gene is on the X chromosome and not one of the other numbered chromosomes. And that's important because as women, you can see we have 2 X's. So if we inherit a dystrophin gene change on one of our chromosomes, we have a second backup copy, which is why we're typically just carriers. Whereas men, whatever they inherit on that 1X that they have, it's going to be expressed because that Y chromosome has different material. So that kind of helps explain why in X-linked conditions, not just Duchenne, but all X-linked conditions, it's typically the men who are more affected. So back to the gene. So the DMD gene or dystrophin gene, like I said, is on the X chromosome, and it's actually one of the largest genes in the human body. It has 79 exons. Maybe you've heard of the term exon. If you haven't, all an exon is just a small segment within the gene. Some genes are very small and only have a few exons, but the dystrophin gene is huge and has 79 exons. The introns you see there are the pieces of DNA in between the exons. In the past, it was thought that these were primarily just junk DNA, but we now know that's not entirely true. But the introns are spliced out. So all of those introns are spliced or cut out And all of the exons have to line up in perfect order in order to form the dystrophin protein, at least a functional dystrophin protein. This next slide you maybe have seen before, but this is actually all 79 exons lined up together. And you can see it's like a puzzle. They have to fit together just perfectly in order to get that normal functioning dystrophin protein. So there are many types of changes that can actually occur in the dystrophin gene because it is so large. It can acquire actually thousands of different types of gene changes. You're going to hear different terms. I'll sometimes say gene change. That's the same as a mutation, and that's the same as a variant. If you've had testing done recently, they tend to use the word variant more now. And you may see words such as pathogenic variant. Pathogenic means that it's a known disease-causing gene change. Benign variant obviously means they know it's a benign change and does not cause disease. Another term you might see on reports is a variant of unknown significance. Those are less common in Duchenne, but there's still some that are showing up. And certainly if you have a report that has something like that, please reach out to us. We are happy to explain what that means. But basically all of the changes in the Duchenne gene fall into three large categories. There's the large deletions. That's when an entire exon within the gene is deleted or several exons deleted. And that's actually 60 to 70% of boys with Duchenne. So it is the large majority of gene changes that we see, even higher for Becker. Duplications are when an entire exon is duplicated or repeated. That's less common. And then the third category are the point mutations, or the very small changes. And that's when a change is happening down at that letter level with the A's, T's, G's, and C's. So it's not involving an entire exon, but it's actually just a very small change within an exon. And that accounts for about 15 to 30% of Duchenne cases and even less for Becker. So this chart here kind of helps show what some of these different mutations mean. If you look at that top sentence, the old dog ran too far, you can think of that as the gene and each of those words as an exon within the gene. So if you have a word or an exon missing, that's a deletion. If you have a word or exon that's repeated, that's a duplication. That third one down is showing a nonsense mutation. The nonsense mutations are actually the most common point mutation that we see in Duchenne. And that's when there's often just one letter that has changed. But in doing so, it creates what we call a premature stop in the production of the protein. So literally the change makes no sense and the protein just stops being made. And what's made is a very short protein that is typically degraded within the cell. So that is nonsense mutation. The next line is showing a missense. A missense is when there is just a one-letter change. So instead of the old dog ran too far, you get the old dog ran too fat. So you get basically a different word that makes no sense. And on the protein level, what happens is you get a new amino acid or a different amino acid in that position, and that can make the protein not functional. And then the last one there is an insertion, and that's basically when there's an extra word or exon inserted in. But by far the most common mutations you'll see are the deletions, duplications, and nonsense mutations. And I do want to point out, if you have your child's report or your own report and you have any questions about the type of mutation you have or you don't understand what it means, that's what we're here for. We have 3 genetic counselors now at PPMD. We love to look at genetic test reports. We are here to help you, so you can email or fax us the reports, and we can review those with you over the phone and help you understand what that means and what it means for your family. So really quickly, the next few slides, I just wanted to touch upon the reading frame rule, which some of you may have heard about, especially if your child has a deletion, because deletions are typically categorized as out of frame or in frame. So this picture is showing what we normally want to happen. As I said before, you have this sentence made of words or the exons, and you want all the exons to line up perfectly so you get a sentence that makes sense. The old dog ran. So that protein would be a normal functioning protein. Everything worked great. In Duchenne, what typically happens is you have an exon deleted, and then the remaining exons cannot fit together properly. And so you get a sentence or a protein that makes no sense. The old ran. And typically what happens there, the protein makes no sense and can't do its job and is often just degraded in the cell. So that is an out-of-frame deletion because you're messing up the reading frame of the protein. With Becker, what happens, you can have a deletion, but those remaining exons can still fit together properly and still make some sense. So here you get the dog ran. Well, that still makes some sense. You get a protein that's still partially functional. That's what we call an in-frame deletion because the reading frame isn't totally knocked out of whack. So in summary here, an out-of-frame deletion will typically produce no dystrophin and cause Duchenne, which is the more severe presentation, whereas an in-frame mutation or deletion will result in some partially functional dystrophin protein, and that's why you typically get the more milder Becker presentation. Now, this reading frame rule is not perfect 100% of the time. There are some exceptions to that, and we're happy to talk with you if you fall into any of those categories. I also want to point out that this reading frame is actually the premise for exon skipping, which I'm not going to talk about because there's a couple industry presentations online for you that will go into more detail about exon skipping. But the whole point of exon skipping is to take an out-of-frame deletion and turn it into an in-frame deletion. Excuse me. So if you can turn an out-of-frame deletion into an in-frame deletion, then you would hope that for that child you could create a more mild course for them, a more Becker-like course. So that is really the whole point of exon skipping. So I encourage you to listen to those talks. I know both Sarepta Therapeutics and NS Pharma both have talks for you that Kaylin emailed out to everybody. All right, so I'm going to jump back a little bit to our chromosomes and the X chromosome and why that matters in terms of inheritance, which I kind of alluded to before. But with X-linked inheritance and with that dystrophin gene being on the X chromosome, if you're a woman who carries that on one of your X chromosomes, then every time you have a child, you have a 50% chance of either passing on your X with the normal functioning copy of the gene or a 50% chance of passing on the X that has the dystrophin gene mutation. If you think about a woman having boys, each time she has a boy, there'd be a 50% chance that he would be affected with Duchenne because boys don't get a second X chromosome from their dad. They get a Y chromosome from their dad. So whatever boys get on that X from their mom is going to be expressed. Whereas if a carrier mom has a daughter, there's a 50% chance her daughter would be a carrier like her because daughters get their second X from their dad. They don't get their dad's Y chromosome or they would be boys. So it's really important to know if you're a carrier. And I'm going to talk more about carrier testing here. So I think there's a lot of misconceptions about who is a carrier and who isn't. I think some mothers think if they have a child with Duchenne that they're automatically a carrier, and that's actually not the case. About 30% of the time, mothers are not carriers. 30% of the time, or 1/3 of the time, that gene change started new in their affected son and was not inherited. Now, 2/3 of the time, it was inherited. And of course, this is just for families where there's one new affected boy. If there's a family history, like if the mother's brother or uncle had Duchenne, then of course she would be a carrier. So the chances I'm talking about here are for families where there's a brand new case in the family. So it's really important to get carrier testing done. If there's any women listening, it's important to know if you're a carrier, not only for family planning reasons and what that would mean for your future pregnancies, but also other women in your family who could be at risk to be carriers. But it's really important for your own health because, as I mentioned before and has been mentioned by other people, we know that women who are carriers can actually have some signs of Duchenne. Many do not, but it is possible to have even some significant cardiac issues. So you need to know if you're a carrier so that you can be appropriately screened. Okay, so I've been talking a lot about genetics and why genetics is so important. It is important for many reasons, obviously, to get the correct diagnosis. It's really the gold standard for diagnosis now with Duchenne and Becker. Years past, it was done just through CK and muscle biopsy, but now genetic testing really needs to be done, and it's so important not only for the patient but for the rest of the family, because once we know the gene mutation in the family, we can easily do carrier testing on the mom and anybody else who would need that. But it's also really important now for research and development and all of the therapies, because so many of the therapies being developed or that are already approved are mutation specific. So you really have to know what your mutation is and understand what that means. I'm not going to spend a lot of time talking about some of these mutation-specific therapies because, again, the industry presentations that are saved for you online go into a lot of detail, but the two most common mutation-specific therapies that you'll hear about are the nonsense mutation read-through for people who have a nonsense mutation. I encourage you to listen to PTC Therapeutics talk because they're going to talk about their therapy, which is called Ateloran or Translarna. And of course, exon skipping. Sarepta Therapeutics now has 2 approved drugs for exon skipping. They have Exondys 51 for skipping of 51 and Vyondys 53 for skipping of 53. And then NS Pharma is also working in the exon skipping area. So I encourage you to listen to both of those talks. And then, of course, gene therapy is in trial now and holds a lot of promise, and there's a lot of excitement around that. So, again, if you have questions about your child's mutation and whether or not they qualify for any of these mutation-specific therapies, please reach out to us, and I'll have my email here at the end of the talk, and I'm sure Kaylin will share it with you as well. But I do want to point out that if your child doesn't fall into one of these categories that qualifies for the mutation-specific therapies, that is okay because there are a lot of therapies in development, a lot in the pipeline that are not mutation-specific, and that would apply to all boys with Duchenne, regardless of your mutation. So I really do want to stress that for you. So the role of the genetic counselors here at PPMD, I think I mentioned there's 3 of us now, which is really exciting. We're here to answer your questions about genetic testing, about what trials your child may or may not be eligible for. We also run a few of the programs within PPMD, the Duchenne Registry, which I'm going to talk about in a few minutes. And then we actually are piloting a newborn screening program in New York State, and one of our genetic counselors, Jen Ely, is overseeing that. And then Jen also runs our DECODE Duchenne free genetic testing program, which I really want to highlight. This is a great program. We've been providing free genetic testing since 2013. And we've been providing this for Duchenne and Becker, and it's with support from Sarepta and PTC. So I really want to thank our sponsors because we would not be able to do this free testing without their support. So thank you, thank you. But if any of you listening have a child who's not yet had genetic testing, or maybe your child's a bit older and the testing was done many years ago and might need repeated, we're happy to look at that to see if the testing should be repeated using newer, more robust methods. It's really easy to qualify for our program. I mean, we'll take somebody with a new diagnosis or somebody who was diagnosed years ago, And we also can now do free testing for females, free carrier testing, which has been a great benefit for the community because so often women have trouble getting their insurance to cover carrier testing. So it's a very simple process. Your doctor does need to go onto our website and do an online application, but it's actually pretty quick and simple. And if you have any questions, reach out to me. I can help you get that started. But all of the testing is performed at Perkin-Elmer Genetics. We've tested, I think this number is actually higher now, closer to 1,200 individuals have been tested free through our DECODE program, and between 200 and 300 carriers. So it's a really great example of how advocacy can work with industry to really help benefit the community. So please take advantage of this program if you need it. All right, in my last few minutes, I want to highlight the Duchenne Registry. I'm actually the director of the registry, so this is very near and dear to my heart. The registry has been around since 2007. It used to be called Duchenne Connect, so maybe you're familiar with that name. But this registry is a self-report registry for Duchenne and Becker, as well as carrier females. Again, we started many years ago. We actually have over 5,000 registrants in the registry. It is international. We have over 100 countries represented. We have over 12 years now of longitudinal data on those patients that have been with us since the beginning. And we've helped to recruit for over 100 clinical trials and research studies. And that's really a great benefit for the families because so many trial sponsors or researchers come to us to recruit, and we can search the registry to see who appears to match criteria and then email you and let you know that your child or that you appear to match criteria for this trial, and this is who you contact for more information. So it's a really great benefit. So the biggest news with the registry over the past year is that we have transitioned to a mobile app platform. We were previously on a web platform, but from talking with a lot of community members, we really felt like we needed to make the registry easier to access and just more user-friendly. So because everyone's on their smartphones now, we have transitioned with the help of Thread Research to develop this new registry app. So hopefully some of you have already joined our app. It's very simple. When you get into the app, you'll see some welcome screens that talk about the goals of the registry. You click Join Study. You'll move through our consent process. Even if you've been in the registry in the past, you do need to re-consent in our app, but it's quite simple. You just swipe through the consent education screens and actually sign right on the screen. Once you're fully registered and consented, you'll see that main screen there to the left, which is the activity screen, which lists all the surveys. And again, if you're a past registrant, you'll recognize a lot of these surveys. They're very similar to the surveys we had before. There is one new one there listed at the top, the About Me survey. That's really important to take because that's where we get all your basic demographic information. So these surveys are really simple to move through. We tried to keep them as short as possible. You basically click on the survey. You get one question per screen. You click on the correct answer, move to the next one, and it's actually a pretty simple, streamlined process. So why should you join the registry? I think Pat mentioned this earlier. This is a great way to be a citizen scientist. And what does that mean? It just means you can really help advance research and move treatments and improve care in the community just by sharing your data through the registry. So you don't have to participate in a trial or a research study if that's not possible for you. Just by joining the registry and downloading the app and participating, you are really serving as a citizen scientist. And of course, as I mentioned before, if you are interested in trials and research studies, it's a great way to stay informed about that, as well as new drug approvals. And of course, just by sharing your data, you're really helping to gain a better understanding of everyone with Duchenne and Becker and carriers, because this data really is used by researchers around the world. So it's, your data isn't just sitting here not being utilized. It is very well utilized, and it's so important. Finally, I do just want to point out, if you would go to the Apple App Store or Google Play Store and download the app, to move through the app, you do need this activation code, which if you're a previous registrant, it was already sent to you. If you can't find it, please contact me. Brand new registrants, though, you actually start on our website, so DuchenneRegistry.org. You click join, you'll see this pre-registration page. All you have to do is enter your email, and then you will get an email invitation from Thread Research that will contain your link to download the app as well as your activation code. So I know I ran through all of that really quickly. If you have any questions at all, oops, my next slide is not showing. Let me back up. If you have any questions at all, please reach out to me, this coordinator at parentprojectmd.org. That reaches our genetic counselor team. As I mentioned, it's myself, Jen Ely, and Nikki Armstrong, who just joined us earlier this year. If you want to reach me directly, my email is Ann, A-N-N, @parentprojectmd.org. And then that 888 number listed on the screen is a direct line to our registry and the genetic counselor. So please reach out if you have any questions at all. So thank you for listening.
Thank you, Anne. That was really terrific. It is so amazing what the registry has done, and certainly the help for trials that it has provided to so many families. I'm really proud of the work you all have done, and certainly the new technology is pretty amazing as well.